What is life made of? You might think about bones, muscles, or even DNA, but at its core, all life as we know it is made up of tiny building blocks called cells. These cells are constantly growing, changing, and yes, even dividing. This process of cell division, where one cell splits into two identical copies, is called mitosis. It's happening right now all throughout your body, making sure you can grow, heal, and thrive. Imagine a bustling city. Buildings represent cells, and just like any city, new buildings are constantly being built to replace old ones or accommodate a growing population. Mitosis is like the city planner, meticulously ensuring that each new building is an exact replica of its predecessor, maintaining order and function within the city. Without mitosis, our bodies would be stuck in time, unable to repair damage or grow new tissues. We would be like a city with no construction workers, slowly crumbling without a means to rebuild. Mitosis, in its elegant simplicity, is the engine that drives the constant renewal of life. The cell cycle consists of two main phases, interphase and mitotic phase. Interphase is the longest, with 3 subphases. The G1 phase is the first gap phase, where the cell grows and synthesizes proteins and organelles. In the S phase, DNA replication occurs, resulting in 2 identical copies of each chromosome called sister chromatids. During the G2 phase, the cell continues to grow and prepare for mitosis, ensuring all components are ready for division. Next comes the M phase, or mitotic phase, a crucial period where a single cell divides, producing 2 genetically identical daughter cells. The M phase is further divided into 4 stages: prophase, metaphase, anaphase, and telophase. Prophase, chromatin condenses into visible chromosomes. Metaphase, chromosomes align at the center. Anaphase, chromosomes are pulled apart toward opposite ends of the cell. Finally, in telophase, 2 new nuclei form, completing the division. Each stage is crucial, ensuring that the resulting cells are perfect copies of the original. As we have seen, mitosis is a continuous process. Now let's take a closer look at its stages. You can think of it like baking a cake. You wouldn't just throw all the ingredients in at once, right? There's a specific order to follow, a recipe. Let's dive in. During prophase, the nucleolus disappears and the chromatin condenses into chromosomes. This condensation ensures that the genetic material is prepared for division. Spindle fibers begin to form from microtubules extending from the centrioles. This spindle fibers plays a crucial role in capturing, aligning, and ultimately separating the chromosomes. As this happens, the nuclear envelope starts to break down, allowing the chromosomes to interact with the mitotic machinery. Next, the chromosomes line up along the metaphase plate, also called the equator, an imaginary plane equidistant from the 2 spindle poles. This precise alignment ensures that each sister chromatid is attached to spindle fibers, setting the stage for accurate chromosome segregation later on. During anaphase, sister chromatids separate and move towards opposite poles of the cell. This process begins when the centromeres divide, enabling each chromatid to become an independent chromosome. Spindle fibers attached to the chromosomes shorten, pulling them apart, while non-kinetochore microtubules elongate the cell, preparing it for division. Telophase is the final stage of mitosis, where the chromosomes reach the poles, the spindle disassembles, the nuclear envelope reforms, chromosomes decondense, and nucleolus reappear within the newly formed nuclei. This stage prepares the cell for cytokinesis, the division of the cytoplasm, ensuring the formation of 2 genetically identical daughter cells. Mitosis might seem like a microscopic phenomenon, but its impact is visible all around us. Think about a child growing taller. This rapid growth is fueled by millions of cells dividing in their bones, muscles, and organs. Or think about a wound healing, seamlessly knitting itself back together. That's mitosis at work, rapidly producing new skin cells to repair the damage. Even plants rely on mitosis for their growth and development. When you plant a seed and watch a tiny sprout emerge from the soil, you're witnessing the power of mitosis. The seed contains a tiny embryo, a cluster of cells that rapidly divide and differentiate, eventually forming roots, stems, leaves, and ultimately an entirely new plant. From the tiniest wound healing to the tallest trees reaching for the sky, mitosis is the invisible force driving growth and renewal in all living things. The discovery of mitosis marked a turning point in biological science. In the late 19th century, German biologist Walter Flemming meticulously documented cell division, becoming the first to observe and describe mitosis. He also pioneered the study of chromosomes. Born on April 21, 1843 in Sachsenburg, Germany, Flemming studied medicine and later developed a passion for microscopy. His curiosity about cell function led him to his groundbreaking discovery. In the 1870s, Flemming used newly developed aniline dyes to stain cells to observe their internal structures. These dyes highlighted chromatin, allowing him to witness a never-before-seen process: the condensation, alignment, and separation of chromosomes. Flemming meticulously documented his observations, naming the process mitosis after the Greek word mitos, meaning thread, due to the thread-like appearance of chromosomes. His detailed sketches laid the foundation for modern cell biology, and his microscope is still preserved today at the University of Kiel's anatomical department. Today, Flemming's work remains crucial in genetics, medicine, and cancer research. Understanding mitosis has helped scientists develop treatments for diseases and explore the complexities of life at a cellular level. The significance of mitosis extends far beyond the laboratory. It is fundamental to the growth, development, and healing of all living organisms.